Hello everyone, and I hope that you're doing well. Today we're doing another review of a CSEC Biology paper. This is a paper 2 from the year 2021, and this is a May-June paper. Now, please remember, if we start the examination, you should read through the instructions carefully, and if you should have any questions or queries, please clarify those with the examiner before you begin. Now, you should also remember you should not open the booklet until you are told to do so. Now let's jump into section A, question number 1. And this question reads that name the 2 raw materials and state the 2 conditions that are necessary for photosynthesis. And so by now you know that the raw materials are water and carbon dioxide, and the conditions is light must be present. And so I have here a lit environment, and chlorophyll must also be present in the leaf. All right, and so let's go into part B, section 1. And so part B1 reads that figure 1 is a drawing of a leaf. It's on the grid provided on page 5. Make a drawing of the leaf that is twice the size of figure 1. And so what I've done is to measure the grid along the leaf and also across the leaf. And so that will give me a general idea of how big the leaf is. And so when you're measuring, when you're going across a matter of fact, you measure the widest part. And so on my grid, I will show you I have 2 red lines here just for guide in terms of what I've done. So I measure across and along the leaf, and then I draw my leaf in that space. Again, the number of grids going across and also along the leaf, that is very important. And it's also important also if you should calculate the magnification of the leaf. All right, the red lines aren't necessary for you to actually have. It's just for guide. And then the next part of the question, let me just go back up. It asks us now to, on the diagram drawn on page 5, label each of the following parts of the leaf. We're going to label apex, lamina, midrib, and petiole. It is a 3 marks question. However, you have 4 things to label. Again, follow the instructions and also look at your marks as well. And so the very tip of the leaf is called the apex. You have the midrib, which is the center vein. And then you have the lamina, which is the general area of the leaf. And you have the petiole, which is pretty much the stem of the leaf. Again, you should also know the side veins and also know the margin of the leaf as well, or the leaf, and also the leaf blade. And so those things you need to know for upcoming exams, just in case they may ask you. Now for section C, or part C, it said the leaf shown in figure 1 on page 4 is from a plant that grew in a very windy conditions. And it suggests one way in which the leaf is adapted for each of the following functions. And so for gaseous exchange, if the wind is moving fast or very windy, then most of the stomata are located on the leaf or on the lower side of the leaf. And the purpose for that is to capture the upward moving gases. All right. And so also it must have a large surface area, because if the gases are moving fast, then a larger surface area will increase the efficiency or the effectiveness in capturing the fast moving gas. The leaf must also be thin to allow easy diffusion of gases into the leaf. All right, and the next one here is saying preventing excess water loss. And so one of the things that could happen here, they could have small stomata compared to other leaves. Because of course, higher wind will increase the rate of transpiration. And so smaller stomata is good. Fewer stomata is also good. A small leaf is also good, and narrow leaves is also good. Anything to reduce the number of stomata or surface area, that will also prevent excess water loss or reduce transpiration. So anything along that line, once you state it properly, you will earn your marks. Now for part D, it's a green plants can produce food by photosynthesis. Suggests 3 reasons why there is still a need for them to store food. So why plants store food even though they can photosynthesize? And one is to produce energy readily as needed. So even though you can make it to store it, you can get energy from the store food whenever you need. A second option here is having food available in case of low light. So when there's no light, the stored food can be used. And another one here is that having food available in case of low water. So if there's limited water to carry out photosynthesis, then definitely that will help. The store food will help in terms of providing energy. And a fourth option here is conserving food in the event the stomata are closed to conserve water. So just in case you have high temperature, for example, low humidity, and the plant is not photosynthesizing as effectively, then you can use the stored food as well. All right, and so let's now get into the next part of the question, and we are on part E. And here it suggests 3 reasons why the initial carbohydrate product of photosynthesis, which is glucose, is not stored in green leaf. So this is kind of a blur. I hope you can see it well. All right, so I'm going to read it one more time. And it suggests 3 reasons why the initial carbohydrate product of photosynthesis, which is glucose, is not stored in green leaves. All right, and so one option here is that it is the main production site, hence the space in the cells are needed for production rather than for storage. So since the leaves are producing glucose, right, there's no need to store it there because it is already producing. And so again, the more space I have for production is better. So if you store it, it's going to take up a lot of space. All right, and so the next option here is that glucose is difficult to be stored, and the reason for that, it is easily absorbed. So if you store glucose in the leaves, it will be absorbed by the cells, and if the cells are getting glucose that are stored, then it will not produce effectively. All right, because the leaves will be tricked, in other words, in terms of it has readily available glucose, right? So there's no need to store it there. All right, the third option here is that the leaves can readily use glucose produced from photosynthesis, and this is supposed to be photosynthesis here. So the leaves can easily, can readily use glucose produced from photosynthesis. All right, let's put that in real quick. All right. All right, so let's jump to the next part of the question now. And we're on part F. And for part F, it said describe the test that could be used to determine if green leaf has stored starch. And so the test if starch is present in the leaf. We have done this before in one of the past examination, and I also have a separate lesson for testing leaf for starch. You can check that out as well. And so quickly run through is to boil the leaf until it is soft, and you do that for a few minutes. And then you're going to remove the leaf from the boiling water and place it in a test tube. Then you cover the leaf with alcohol and then boil it until the leaf become white. And the reason for doing that is to remove chlorophyll. And then once you have removed the chlorophyll with the alcohol, then you're going to remove the leaf and rehydrate the leaf, which make it become soft again. Because once you boil the leaf in alcohol, it becomes stiff. And once you have done that, matter of fact, to do that, to make the leaf rehydrate, you're going to put it in the boiling water or the warm water. And then you're going to remove it from the boiling water and place it onto a mat or a piece of napkin or in a petri dish. And then you're going to add a few drops of iodine, and you're going to note the color change. And what you should get is a blue-black color if starch is present. If you do not obtain a blue-black color, then starch is not present in the leaf. All right, so let's go on to the next part of the question, this is part G. And this part states that unlike plants, animals must ingest complex food substances, then break these down by mechanical and chemical digestion. It's explained 2 ways in which the human digestive system is adapted for mechanical digestion of food. And so one is that teeth are present in the mouth for chewing. And so here is the teeth help with tearing, cutting, and grinding of food into smaller portions. And the next thing here is that the stomach is important for chemical and also physical digestion. So this is the physical part here, or mechanical digestion, that the stomach is muscular and rigid to churn food. And it's the churning of food in the stomach is a result of a series of contractions and the contraction of the muscles, resulting in an increase in surface area of the food. All right, so those are the 2 adaptations for mechanical digestion. Now, for question number 2, is to define the term pollination. And pollination is the transfer of pollen from the anther to the stigma of a flower of the same species. All right. And so that is very important. They must include that they are must be of the same species. Now, a point to note here is that the flower could be the same or a different plant of the same species. And if the flower is the same or the same plant, that is self-pollination. And if it's a different flower from a different plant, then that is what they call cross-pollination. Just to mention that in there. So you need to know different types of pollination. And also very important to know also the agents of pollination, all right, such as wind, insect, and so on. All right, for part B, it's to describe how male gametes reach a female gamete in humans. And this is what takes place. A number of things happen here. Is that the sperm cells are present in the seminal fluid that is deposited into the female's reproductive system. And the seminal fluid now provide a medium for the sperm cells to swim. And the second thing you need to mention here is that the sperm cells contain flagella that propel and to generate the swimming actions of the sperm cells. And the sperm cells, they will swim through the structures of the female reproductive system until they reach the egg in the fallopian tube. And once they reach the egg in the fallopian tube, then they will unite. And once they unite, and particular to mention is the nuclei unite. The nucleus from the sperm and the nucleus from the egg unite or fuse together to form what they call a zygote. All right, and so part C is a figure 2, shows the female part of a flower in which the petals, sepals, and stamen have been removed. It's a label each of the following parts on the diagram. You're going to label the stigma, style, ovary, and the ovule. And the stigma is the top part with the sticky. The style is a long narrow tube between the stigma and the ovary. And the style is where the pollen tube will be formed. I need to know the process of fertilization in flowering plant as well. And here we have the ovule and we have the ovary. All right, so now let's jump into the next part of the question. And this is part D. And this part said that explain why a human embryo does not have large nutrient stores, but a plant embryo does. And so I kind of separate them in terms of the two things. And so the human embryo is connected to its mother and receive the required nutrients from the mother. Hence, there's no need to store food. However, now in the plant embryo is not connected to the parent plant. And so because of this now, it needs a reserve of nutrients to aid in growth and development. All right, so it has to be, in other words, self-sustained until, it can make its own food. All right, and then the plant embryo now depends on the stored nutrients until it develops to the point where it can photosynthesize. All right, so again, the plant embryo is not connected to the parent plant, so it depends on the food that is stored in the seed. All right, and that I think that's a 4 marks question. I'm almost sure. Yeah, it's a 4 marks. All right. Now for part E is to suggest 4 reasons why a couple would be advised to use the permanent method of birth control for the male rather than the permanent method of birth control for the female. All right, and so there's a number of reasons. I know this is kind of males will say, boy, no, they don't want to do it. They allow the female to do it because most times anything reproductive is put pressure on the female, to be honest with you. It's a kind of unfair thing, but it is better in male for these following reasons. Is that one is if there's a change of mind, it is easier to extract sperm cells and transfer to the woman. The other reason is that generally in males it is safer, all right, because there's less possible complications to do it in males, and it's also simpler as well. And for all of these reasons, it is also cheaper. And another reason here, it is easier to be surgically reversed. So if you need to reverse it, you kind of tie in. Well, it's not tying to number 1 because number 1 is just to extract the sperm directly. But this one, if you should reverse it on a surgical means, then it is easier to do so. And in some way, it is technically more effective. The effectiveness of them in terms of how reliable they are pretty much equal. But in terms of effectiveness, in terms of less complications, easier to do, it's more guaranteed than even the female, because based on the different tubes and possible complication, then the effectiveness of it is more in that terms, as I put that in red, because that can be debatable. As again, they are reliable pretty much the same way once they have done properly. All right, because it's pretty much the same thing or the same concept. All right, just that one is male, one is female, one is the fallopian tube, one is the sperm duct. So again, that one can be argued, and depends on how you explain that one too. All right. Now number 3, it's a figure 3 is a diagram of a terrestrial food chain. And so here we have a terrestrial food chain, and we notice we have a wildflower, we have grass, mouse, rabbit, auk, snake, and you can see the transfer of energy or the flow of food. And so part A is a using the food web illustrate in figure 3, identify one producer. And producer, we have 2 producers here. We have the wildflower and the grass. So we have 2 options there. So any of those will be correct. And predator now, the best suited predator here will be the auk and snake, as they are hunting and killing their prey. All right. And it asks for primary consumers, and primary consumers will be either the rabbit or the mouse. The 3 marks, so one of the listed 2 that I gave will be correct. So it's kind of hard for you to get this section here wrong. Even if you guess it, trust me, you shouldn't be guessing this food chain stuff by now anyway, but I'm just saying it's kind of easy to gain those 3 marks, or at least total of 3. Now for part B, it's a state 2 roles of the producer in the food web. And one is to supply energy to the other organism. That's the main purpose for producers. And a second reason is to convert solar energy to chemical energy, which are usable, which chemical energies are usable by other organisms, because animals cannot use solar energy, so they have to be converted to chemical energy, which they get that from the producer. All right, and so again, you can convert inorganic substances. We're talking about here carbon dioxide and water to organic substances, and the organic substances are usable by the other organisms. So glucose molecules, for example, are taken in, and of course, the glucose molecules can use to produce different substances within the animals. And now we have part C, is to identify one biotic factor and one abiotic factor that would affect the organisms shown in figure 3. And so a biotic factor here could be increase in population of the auk or snake. All right. And so if the population of the auk or snake increase, then the other 2 organisms, which are the herbivores, they will decrease. And if they decrease, then the population of the producers will increase. All right. And the abiotic factors now, if you have less sunlight, so decrease in sunlight, decrease in water or carbon dioxide, once any of those change, then it can affect the rate of production of food, and so therefore the energy flow will also decrease as well, putting more pressure on the organisms or increase the competition for food, and hence you'll have decline in population. As a matter of fact, all the organisms that follow the producer or come after the producer will be affected if the producer decrease in energy production or decrease in numbers. For part D, the state explain why there are fewer organisms at the top of the top than at the bottom of the food web. And the basic reason here is that as you go along the food web, energy decreases. So energy decreases from bottom to top. And energy decreases by 10% technically in a food chain. All right, but generally, as you go up the food web as well, energy decreases as well. And so the next thing here is that at the top, organisms tend to be larger and require more energy to carry their functions. And so if there are more consumers than producers, then definitely some of them will not have food. They have to compete for the same thing. So as they go up the food web, they must have less number to decrease competition for food or resources. All right, and so hence energy can be used effectively if you have less competition for the same resource. Now, for part 2 of this section, is to discuss 2 possible consequences of the auk becoming extinct in the ecosystem in figure 3. And so if the auk becomes extinct, what will happen is the population of rabbits and mouse will increase. And you can go back to you can just rewind and go back to the food web, and you see exactly what I'm talking about. So again, the population of rabbit and mouse will increase for sure, and so they will have less predators feeding on them. That's the reason why there's an increase. And another thing here is that the population of the wildflowers and grass will decrease. And the reason for that is because if you have less auk and the population of rabbit and mouse increase, then there'll be more herbivores. And so if you have more herbivores, demand for energy directly from the producers will increase. Thus, more of the producers will be consumed. And so that's the reason why you have less producers. All right, so the one in red will be the explanation for the statement in blue. Now for part E, which is the last section for this question, is to suggest 2 reasons why decomposers are important organisms in all food webs. And decomposers are important. One, because they recycle nutrients, they convert organic molecules to inorganic molecules, and plants use inorganic molecules. And also they aid in the health and development of the producers, again, because they supply nutrients to the producers. And also they help to convert waste into useful substances. And all of these will be directly contributed to the producers. And once you have more producers, healthier producers that can produce more food and energy, then hence you'd have a more healthy ecosystem as well, and even more diverse ecosystem. Now, section B, question number 4. Now, the first part of this question is a figure 4 is a labeled diagram of a nephron, and you can observe the different structures of a nephron. And then the first part of the question now asks us, is a use figure 4 to describe how urine is formed. And so you need to know that urine is a mixture of urea, salts, and water. And so here I give you an outline here. One is that substances are filtered from the blood into the glomerulus of the kidneys. And of course, that will go into the Bowsman capsule, as you know. And the substances will include water, salts, urea, and glucose. And then now this is supposed to read here, the filtered. So let's put that there. So the filtered substances travel through the tubules of the nephron. While the substances passes through the tubules, glucose is being absorbed into the capillaries. And so glucose will be reabsorbed back into the blood. And so at this point now, at this point of the collecting duct, all of the glomerulus, all of the glucose, I should say, I keep glomerulus in my head. So all of the glucose would have been reabsorbed by this point. And so the next point I have here is that in the collecting duct, only water, salts, and urea are left. And so a mixture of the water, salts, and urea in the collecting duct result in the formation of urine. All right. And so even though glucose is being filtered, it will be reabsorbed back into the capillaries along the tubules of the kidney. Of the nephron of the kidney, to be more specific. All right. So next part of the question here now. It asks us now to explain why there is a high concentration of glucose in the urine of a diabetic patient. And one does make a pretty good note here is that in diabetic patient, the glucose is not effectively used by the cells or converted to glycogen for storage. So that's the main thing is happening in a diabetic person. And so the reason why now it will be high in the blood is, these reasons that follow is that once that glucose concentrations are in the blood, and therefore it is treated as a waste product. So since a lot of glucose is in the blood because the body cannot use it or store it effectively, and so because now what will happen here is that due to the high level of glucose in the blood, a lot of it is being filtered by the glomerulus in the Bowsman capsule and into the kidney tubules. Now what is happening here is that most of the filtered glucose is unable to reabsorb because you have so much glucose there. And matter of fact, the body is trying to get rid of some of it. And glucose cannot go through the skin like that so easily because, again, to move through the capillaries, glucose is kind of a large molecule, right? But glucose is easily filtered through the glomerulus. But glucose does not go through other capillaries generally, all right? Moving out of the body. It normally go into cells, all right, just to make that point. And it said most of the filtered glucose now is unable to reabsorb by the nephron and hence is present in high concentration in urine. All right? Because again, so much is passing through, hard to reabsorb all of it or effectively absorb all of it. So some will definitely be in the urine itself. All right, and so next part here is a state one similarity and two differences between the method by which the human body regulates blood sugar levels and the method by which the human body regulates the amount of water in the blood. And so here we're talking about two different things. We're talking about control of water and also regulation of sugar. So two different processes. But the similarity between these two processes, controlling sugar and also water, they are controlled by hormones. So we say they are hormone regulated. All right. Also, they are involuntary, which means they're automatically controlled by the body. All right. So they are involuntary actions. All right. So you do not think about it. You have no part to do with it. It does not automatically occur in your body. So those are the two similarities. You ask for one, however. Now the difference is that one is that osmoregulation, excess water is removed from the body. However, for a normal person, to control the sugar level is that excess glucose is stored, not removed, but stored. All right? And it's stored in the form of glycogen, as you should know. And for osmoregulation, excess water is removed by osmosis. And this is controlled by the removal of water is moved by osmosis from the body, a matter of fact, and then it go into the kidneys to remove in terms of urine. However, now for glucose or sugar, the excess sugar is removed by diffusion. So if you have excess sugar anywhere, it moves by diffusion, but excess water is moved from cells or the blood by osmosis. All right. So a matter of fact, also mentioned osmosis is a form of diffusion, as you know, right? But it's diffusion of water. All right, but there are two different processes. All right, and so osmoregulation is controlled by one hormone, which is called ADH, which is antidiuretic hormone. And sugar regulation now is controlled by two hormones, which is insulin and glucagon. A matter of fact, both processes are negative feedback. So that could also be a similarity right here that they are controlled by negative feedback. I could just add that in because both are negative feedback processes. So they are controlled by negative feedback. All right. So that's another similarity right there. All right. And so difference now here, the last difference we'll look at here is that the hormone is produced by the brain for osmoregulation and is produced by the hypothalamus, while the hormone are produced by the pancreas to control sugar. All right. In fact, they are produced, I should say, or secreted by the islets of Langerhans in the pancreas. All right, for part C, and part C suggests two ways in which kidney damage can be prevented in diabetic and hypertensive patients. And so one, they can do regular exercises. And of course, these things will take out waste out of the body to reduce excess buildup. So anything that reduce waste or intake of waste, those things are very important in preserving the kidneys. All right, so regular exercise is definitely good. Your body will use up the glucose, use up the electrolytes readily. That will cause hypertension because it's salt. All right, and also the consumption of less sugar and salt is also important as well. Drinking a lot of water to make these substances move through easily, and also to prevent the buildup of sugar because if sugar concentrations are in the blood, drinking a lot of water will kind of make the blood be at its regular consistency. Take medicines if needed to reduce blood pressure because the blood pressure will build up if you have if you're hypertensive and also if you have a lot of glucose in the blood because the blood not able to move or flow easily, so your blood pressure will be built up over a period of time. And next one is that you do regular checkups and monitoring. And doing regular checkup and monitoring, that will help you know to guide you in terms of what you need to do at certain time, you know. But of course, taking medicine will not be my first choice. We go the natural means first, and the medicine will be the last method. But of course, depends on the conditions as well and how severe the condition is. Now, for question number 5, For question number 5, you said red and white blood cells are important components of human blood. It said use clearly labeled diagrams and illustrate the structure of red blood cell and the structure of white blood cell. And so we have the red blood cell box right here that they provide for us. And so I just gonna draw 2 diagrams for the red blood cell to show different views. And so we have here the red blood cell, which is a biconcave disc, and it has no nucleus, so no nucleus is there. You have the plasma membrane or the cell membrane, and you have a cytoplasm that is filled with hemoglobin. All right, so those are the general structures of a red blood cell. Now, for a white blood cell, now you have the cell membrane, you have a nucleus. A nucleus is present, and I draw 2 types of white blood cells here. The first one, it is lymphocyte, and the second one is phagocyte. That is one on your right. All right, and so if you notice the abnormally shaped nucleus for phagocyte, and the purpose for that is to change shape for engulfing foreign objects or bacteria. All right, and we have the cytoplasm as regular cell. All right, now let's go to the next part of the question. For this part B is to explain how any 2 features of the red blood cell ensures that it's suited for its major functions in the human body. And so we're going to talk about the different features here. No nucleus. So if there's no nucleus, it allows them to carry out more hemoglobin due to more space. So the absence of nucleus is really creating more space for more hemoglobin. And also it allows them to change shape easily as well because of the absence of nucleus. And once they're able to change shape easily, then they can pass through the capillaries more effectively and easier. Now they're also biconcave, and being biconcave make them easier to travel through the red blood cells as well, or to pass it through the blood vessels. I say red blood cells, but blood vessels. A lot of hemoglobin as well, present a lot of hemoglobin in the red blood cells, and this allows them to pick up and transport oxygen easily and effectively. All right, and so let's go into our next part of our question right here. And for this one, it said the ABO blood grouping system is determined by antigens on the surface of the red blood cells. And if you want to learn about the ABO blood group system, I have a series of lessons on that in terms of all different antigens that are present on the red blood cells to make them certain blood type and so on. So you can go through those and you'll see the lessons that are presented on that. All right, so here it said an individual now who has one parent with heterozygous genotype for blood group A and the other parent with the heterozygous genotype for blood group B may have a blood group phenotype that is different from either parent. So with the aid of a genetic diagram, explain how an individual may have a blood type that is different from either parent. And so here now, I just kind of lay out that the genotype of parent 1 would be AO and the one for parent 2 will be BO. Again, they're heterozygous A and heterozygous B. Now the gamete that you also need to learn to practice how to determine gamete. Gamete comes from the genotype, right? Well, once it's separate the genotype, then you get the gametes pretty much. All right, so parent 1 can only produce A and O gamete. Parent 2 can produce B and also O gamete. And so once I draw a Punnett square now, this is parent 1 with gamete A, gamete O, and then parent 2 with gamete B, gamete O. And these will be the genotype now of the offspring. And so we have a AB, we have a BO, we have a AO, and also OO, which is thus O. All right, and so point to note is that the AB and the OO, which is thus O group, are different from either parent. All right. So one parent again was AO and one parent was BO. So these two here, which is the AB and O, they are different from the parents. And so to explain it, the parents are AO and BO respectively and can have children with AB and O group or O blood group type, which are different from either parent. All right, so that's just the answers right there. For 5 marks, the diagrams with explanation will earn you 5 marks easily. Now, for question number 6, which is our final question. Here it said now outline Darwin's theory of natural selection. And you can actually outline this in two ways. And so the general one, you have overpopulation, overproduction, I should say, and overproduction of offspring, that is. And so organisms produce more offspring than needed for the most part. And then because of this now, you have competition. And once there's a competition, the organism will compete for food and resources. And due to variation, organisms have different physical traits. And that's what variation is. Even though they're the same species, they are phenotypically different. And so what will happen here, because of competition now, you have adaptation. And organisms with favorable traits will survive and reproduce. And so those traits now are passed on to offspring. And so another way to kind of outline Darwin's theory of natural selection as well is also this. You have an environmental pressure. So any changes within the environment, and then because of the changes within the environment, due to difference among the organisms or variation, then that will lead to adaptation, which means the fittest trait will survive and able to pass on. All right, so any of the two pretty much will be good for you to earn that nice 2 marks. All right, I just give explanation for it. It didn't ask me explanations to outline it, but of course you can outline with explanation. There's no problem doing that. Now for part B is to explain why the developing sorry, let me go back over that. I think I missed something. Is to explain why the developing of antibiotic resistance by bacteria is considered to be an example of natural selection. And so the outline of this is that you have an introduction of the antibiotic. That's the first thing that will happen. And the antibiotic now will create a change in the normal or natural environment of the bacteria. And so here I say the antibiotic create a change in the environmental condition. And so here you have what you call added pressure. So pressure is added to the bacteria. And now due to variation, some bacteria are different. And so variation in traits, they have different traits. And then some will be resistant, some will be non-resistant. And so the bacteria now that have favorable traits that allow them to resist the antibiotic will survive and produce bacteria with that antibiotic resistant trait. All right. And so since variation leads to adaptation, lead to the continuation of a specific species or a new species, then definitely it is considered as a form of natural selection. All right, let's go to the next part of the question, or part C. It states that a farmer wishes to use artificial selection to alter the characteristic of his crops. Suggests 2 ways in which artificial selection will benefit the farmer. And so one, because of artificial selection, which is selection done by the farmer, it is done by man. And so what he will realize over a period of time is better yield. And better yield could be faster production, better quality of crops as well. All right. And also it may have more profit. It may have reduction in undesirable crops because he will choose the ones that he desire or better. And so the ones that are so good over time, they will disappear, or he will not have them anymore in his farm. And a fourth reason here that he have better, cheaper pest control method. And let me kind of explain this to you because this one kind of tricky depending on how you state it. When I say better or cheaper pest control method, is simple this. If you choose a particular crop that have a resistant to a certain pest, then you're going to have a whole field of crops that can resist a certain pest. All right. And so over time, you have no need to spend money to control a certain type of pest. Or you may even select other plants as well to biologically control certain pests. So again, that is also a means of artificial selection. Choose the characteristic that can survive a certain environmental condition or to benefit what he desired. All right, and so let's go to part D. This part is a little bit sketchy, so let me just kind of go through this. It's relatively slow so you can catch this. And this one say a species of toad which carry a toxin was introduced on an island which was inhabited by iguanas. It said the iguanas ate mainly toads. It said many but not all of the iguanas died when they ate the toads. It said several years later, biologists examined the iguanas for any changes and observed that the length of the tails of the adult iguanas increased by 10%. And this suggests 3 possible reasons for the increase in tail length observed in these iguanas. And a point to note, because I just put natural selection here because this is due to natural selection. The introduction of toads with toxins will kill some of the toads and only have a few remaining. So the one remains is really the one that remain is really by natural selection. And here now, point to note why we may see longer tails, and this I think is 3 marks. And here it said now the surviving iguanas carry the allele for long tail, and so hence they pass on that trait to their offspring. So that's one possible reason. The next reason is that the toxin could cause mutation in the tail because chemicals and any changes also could lead to mutation. And mutation is a random thing, but you can have triggers such as chemical triggers, environmental pressures. Different things could lead to mutation. All right, there's no specific reason for it or no specific cause, but it's a random process. Now, next thing is that the iguanas may prefer to mate with those with long tails. Hence, over time, only that trait is seen. Another possibility here, just to make a note of this, and I kind of break this one down in different parts, is that more toads equal to more nutrients. And so, if you're looking at toads versus iguana ratio, you may have more toads than iguanas. And so, since some of the iguanas died, then you have a smaller population of iguanas over time, well, in that period of time. And so, you have, because of the killing of some of the iguanas, you may have less competition as well. And since you have less competition for food, then you have more nutrients available for growth and development in those iguanas so they can develop into full size. All right, so this is another way to explain a reason here. All right, and let's go to this part here now. And this part of the question states that one of the biologists suggests that the iguanas with longer tails were a new species, that a matter of fact. So it's a new species right here. So the one with longer tails are a new species. I'm just gonna highlight that real quick. And it said, but another biologist argued that the species is not determined by different physical features. And then it was later concluded that iguanas with longer tails were not a new species. Provide an explanation to support this conclusion. So we're saying that they're not a new species. So once there's a new species, once it's not a new species. But the end result is that it was concluded that, hey, this is not a new species. So behind this, now, the reason behind this is that all iguanas were able to interbreed before the introduction of the toads. So if they were able to interbreed before the introduction of the toads, it means that they all belong to the same species. Hence now, they were same species as mentioned. However, if the introduction of the toads only cause those that are resistant to the toxin, which are the long-tailed iguanas, to survive over the non-resistant iguanas, that doesn't make them different, right? It only means that now only this trait, long-tailed traits, are present, and so they only mate now. And again, what could happen here too? Why didn't you see much long tail? Because long tail could have been a resistant allele. A recessive allele. And if it was recessive, you won't see much of it. But however, what if the recessive allele are the only surviving iguanas because they are the one that resistant to the toxin? Then the recessive allele now could mate with recessive allele and produce that type of trait. So there's a number of things that you can explain, a number of ways you could explain it right here as well. So you can mention it in terms of the allele being recessive to begin with. So hence, at the beginning, there were not a lot of long tails. However, recessive can now mate with recessive, and then you will see more of that trait present in the current population of iguanas. Now, for part E, it said describe two situations which may lead to the evolution of new species. And so there are a number of reasons here that can lead to the evolution of new species. One is by mutation. Again, it's a random change of genes or DNA mutation that are passed on to offspring. And then next one could be here the separation of species by geographic, so you call geographic isolation. And organisms may separate based on habit or location and adapt to the different environmental changes. And once they adapt over a long period of time because of adaptation and changing behavior, they may no longer able to mate with other species. All right, and so we have natural selection as well. As we have been through this paper, we see that a lot. And natural selection is organism with suitable traits within a specific environment will adapt, survive, and pass their genes by reproducing. And so here we have reproductive isolation is another one. An organism may develop different location, time, and other patterns of breeding, resulting in changes within the species. And again, each of these factors here, again, result into one thing is that adaptation must take place. And because of some adaptation, then this pattern that exists may keep passing on to offspring. And again, over time, if they are sexually reproducing organisms, then they can also have other variation again. And so the other variation or new variations may now lead to isolation in terms of interbreeding, and hence new species are formed. All right, and so at the end of this review, and I thank you for watching, and please do well on the examination, and I wish you luck as always. My prayers are with you. And please, if you have not yet, subscribe and also encourage your friends to come over as well. All right, so thank you and take care. See you soon.